I will introduce our speaker this morning. So I want to welcome everyone and thank you for showing up this morning and joining us for Victoria Deren's master's thesis. I'm going to tell you a little bit about Victoria. I'm sure most of you know these things, but just to give her a little bit of an introduction. Prior to joining TSU, Victoria completed her bachelor's at George Mason University, where she studied biology. And while she was taking a course in conservation biology class during her undergrad, that was where she developed an interest in sustainable agriculture. After graduating with her bachelor's, she worked for a few years. And during that time, she gained a wide range of field and lab experience, working as a park ranger, forestry technician, a lab assistant, and joining and participating in the Tennessee Master Gardeners program. So around 2018, Victoria decided she wanted to pursue a master's. She went and interviewed at different schools, and she came to visit us as well here at Tennessee State University at the Nursery Research Center. And we spoke with Victoria, and she came to my lab and we talked about her interests, and specifically she showed a great interest in sustainable and organic production methods and had a keen interest in exploring the use of microbial products for management of pests and disease. And so her thesis project grew out of this interest and a project idea which Dr. Fulya Baisal-Gorel and I had been working on, exploring on the interactions of microbial products. So for the past 2 years, Victoria has worked on this topic and she's done a really great job. She's a pioneer in this area of research, microbial interactions. I think she could tell you that she's learned a lot through the process on what research is and how to overcome some stumbling blocks. And hopefully she'll be a guide for students who want to pursue this area of research in the future. She's currently interning at Corteva AgriScience in Johnston, Iowa, and hopes to pursue a career in the agricultural industry sector. Victoria's presentation today is on simultaneously applied microbial pesticides. And Victoria, you have the screen now.
Thank you so much, Dr. Desso. And I'm going to talk to you today about what happens when you combine microbial pesticides and their impact on the management of disease and arthropod pests. I've always actually been interested in sustainability, and in undergrad, yes, I became interested in sustainable agriculture because that is one part of conservation biology that was never really covered. And one of the questions that's never been answered is if you apply more than one microbial product, do they interact and how do they interact? This is important because the market for microbials is increasing. This is because of the environmental movement and also the organic food movement. It is increasing approximately 10% per year. This means that growers will want to apply more than one product at the same time. There's a lot that we don't know about microbial pesticides, but what we do know is that if you apply beneficial microbes to plant roots, they can change the quality of the plant. This includes the uptake of nutrient availability, altered feeding of pests, induced volatiles that attract predators, and priming the plant so that secondary compounds can be released for protection against pests and pathogens. For this study, we used the following system. We used ornamental pepper because this research can cross over into the food industry. We also looked at Phytophthora capsaicei, a root rot, which was first found in pepper plants. It's an oomycete pathogen that can be transmitted through water. So I also wanted to look at the two-spotted spider mite. It's a generalist pest that can also feed on pepper plants. I also wanted to know how these products may affect Swarovski predatory mite, which is often used to control spider mites and other pests in organic and integrated pest management systems. We use 4 commercially available microbial pesticide products. The first 2 are miticides, Grandivo and Ancora. Grandivo is a bacteria and Ancora is a fungus. And the second 2 were fungicides, which were Root Shield Plus. It is a fungus, and then Stargus, which is a bacteria. I've had 2 objectives in my thesis. In my first objective, we wanted to know if spider mites behave differently when these products were added to the pepper plants. In this objective, products were applied singly to see their individual effects on the mites. My first question was, do these microbial products change how many spider mites there are or where they are located on the plant? Each of the microbial products were applied to 10 replicant plants. Plants were infested with spider mites 1 week before evaluation. Then, once a week over the experiment, we collected 1 random leaf from the top, middle, and bottom strata of the plant, where we counted spider mite adults, nymphs, and eggs. As we can see here, from where spider mites were located on the plant, in the control, we see a pattern where most of the spider mites are located in the middle of the plant, followed by the top and then the bottom. This pattern was repeated in Ancora. However, in Grandivo and Root Shield Plus, more were found in the top canopy and few were found in the middle or the bottom strata of the plant. Now, the most different pattern that we observed was in Stargus, where most were located in the top, followed by the bottom, and then the middle. We don't have an answer for why this is, but we would suggest that repeating this experiment with multiple replicates to see if these patterns are consistent, and if they are, the possible nutrient quality in the foliage. Now, looking at the overall population numbers of the spider mites, you can see that there's a lot of variation, but the highest number are in Stargus, where the fewest were recovered in the miticide Grandivo. We expected to find lower numbers in Grandivo because it's labeled as a miticide, but we did not expect to see such high numbers in the fungicide Stargus. So clearly there were fewer mites on Grandivo plants, and we wanted to know if any of the treatments changed egg laying. So we did an oviposition assay where we added water to the bottom area of a Petri dish with a piece of floral foam. The water acted like a barrier so that the mites did not escape. Then we selected 1 random leaf from each treatment where we cut out a leaf disc. This leaf disc was put onto the piece of floral foam, and 2 female mites were added to the leaf disc. These oviposition assays were incubated at 25 degrees Celsius for a 48-hour period. This was repeated 3 times on day 5, day 14, and day 34. So when evaluating oviposition, only spider mites placed on Grandivo treated leaves showed a reduction in eggs over the 2-day period. Of importance is the 30-day trial. They had reduced egg numbers, despite the fact that it's been 20 days after the last Grandivo application. This shows the long-lasting effects of Grandivo on spider mites. Continuing with our questions about behavior, we wanted to know if spider mites showed any preference for microbial or untreated leaves. We did a choice test assay, where on day 8 and day 9, post-application of the pesticides, we cut out leaf discs from a control and then a treatment, and we put a spider mite in between the middle, and we used EthoVision version 14, a behavioral software, to track the duration of where the mite spent on the Petri dish over a 3-minute period of time. Here you can see a heat map where the mite moved, touched the control disc for a little bit, and then made its way onto the rim. So the only change in behavior was in the Ancora treatment, where mites avoided both of the leaf discs in the center of the arena and spent most of the time on the rim. We wanted to know why mites were avoiding the Ancora leaf discs, so we investigated whether plant volatiles released from the Ancora leaf differed from the others. So we collected plant tissue via flash freeze method, where we use dry ice in an acetone bath to quickly freeze it, and these were stored at -80 degrees Celsius until they could be analyzed. We analyzed the volatiles on a GCMS and compared between treatments. The volatile peaks were identified by matching them to the library. There were no differences observed between any of the treatments, including Ancora. The only major volatiles that were present were the green leaf volatiles. These are basically the smell of grass, and they only appear when the plant has been damaged. So if it wasn't the volatiles that caused Ancora to be a repellent, was it the Ancora product itself? We used another ethovision experiment to do a dilution test from 100% of the application rate down to 0% the application rate. We applied 1 microliter to the center of the Petri dish and allowed it to dry for 30 seconds. A spider mite was then added to the treatment zone and monitored for 3 minutes. In this experiment, spider mites spent less time in the treatment zone when Ancora was present at 100%. The same pattern was observed when Ancora was diluted down to 50%, even though that these were not significant in the 50%, we do see that the same pattern still exists. We concluded that something about the product is repellent to the spider mites. Earlier experiments suggested that Grandivo was toxic to spider mites, but Ancora was not. This experiment was conducted to be sure that no behavioral changes, such as avoidance, were responsible for their survival on the Ancora treated plant. I also wanted to know if the Swarovskii predator was compatible with these products. We had 2 sets of 10 plants. One set was treated with Ancora, the other set was treated with Grandivo. 2 different experiments were performed where mites were exposed to fresh product in a direct exposure assay and to a 3-day aged product in a residual assay. 2 sets of vials were treated with each product. After the product had dried, a treated leaf disc and 1 spider mite were added to the vials for the direct experiment. And for the residual exposure test, the second half of the vials were prepared 3 days later by placing a previously treated leaf disc and 1 spider mite into an aged vial. In both the direct application experiment and the residual, more mites died in the Grandivo, as seen here with the gray line, than they did in the Ancora or the control. The same experiment was conducted with Swarovskii mite, but instead of a leaf disc, a brine shrimp paste was added to the inside cap as a food source. Again, in the direct and residual application, Grandivo had significantly more mortality than either Ancora or the control. This suggests that Grandivo is toxic to both spider mites and Swarovskii. So what did we learn from Objective 1? We learned that Grandivo is toxic to spider mites and Swarovskii mites. Ancora is not toxic to either, but is repellent to spider mites. There did not seem to be any changes to volatiles when the products were applied. For our next objective, we looked to evaluate these products when they were applied together. Just as a reminder, we had 2 miticides, the Grandivo and the Ancora. And we had 2 fungicides, the Root Shield Plus and the Stargus. And then we combined them in all possible combinations of miticides and fungicides. We also had a positive control and a negative control in this experiment. We conducted 3 trials in a small south-facing greenhouse. Plants were at reproductive stage. They were staggered on 2 benches of the greenhouse, and temperatures were set to keep at 77 degrees Fahrenheit to 55 degrees Fahrenheit. Unfortunately, even though temperatures were set in the greenhouse, trials were done at different times of the year and temperatures were not consistent. In trial 1, as seen here, they were consistently hot. In trial 2, they went from hot to cold. And in trial 3, we saw a lot of variation from cold to hot to cold to hot again. Temperature is another variable that potentially affected these interactions. We had already seen how Ancora, Grandivo, Root Shield Plus, and Stargus affected the number of spider mites when applied individually. Now we wanted to see what happens to spider mites and Swarovskii when foliar and soil products were also applied together. In this experiment, Swarovskii were already established on the experimental plants. Plants were infested with spider mites 1 week before the application of miticides. Then 1 random leaf from the top, middle, bottom strata of the plant were collected weekly over the duration of the experiment to find out the population numbers of the Swarovskii and the 2-spotted spider mite. Unfortunately, there were not enough spider mites to evaluate because the predatory mites did a great job at what they should do, which is eat spider mites. But what we did see is that predatory mites survived on all treatments and all trials. We also saw that Grandivo did not have a significant effect, even if it was toxic in the lab. There is a possible pattern for Stargus and Ancora to increase the number of predatory mites. However, this is not consistent in all trials. In order to evaluate the quality of the plant over time, we first looked at wilting. Once a week, we evaluated plants using a wilt scale of 1 to 5, where 1 meant 0% wilt or a healthy plant, and then 5 meant up to 100% or dead. Miticide treatments had no effect on wilt severity. The fungicides decreased wilt severity, but when Grandivo was combined to Root Shield Plus failed. All treatments increased wilt over time, and other than Root Shield Plus, when it was combined with Grandivo, all treatments with beneficial soil microbes had less wilt over time. When Root Shield Plus and Stargus were applied alone, they decreased wilt. When they were combined with miticides and the fungicides failed. Stargus and Root Shield Plus had less wilt over time than any of the other treatments. The same pattern was observed here, where Root Shield Plus and Stargus had low wilting, but when combined with miticides, especially Grandivo, wilting increased. Stargus and Grandivo had the highest amount of wilt, while Stargus had the lowest. We wanted to look at the whole weight of the plant to determine overall growth. Grandivo decreased the weight of the plants when applied alone or with fungicides. Only Root Shield Plus and Stargus, when they were applied alone, increased plant weight. Grandivo decreased plant weight when applied with Root Shield Plus or with Stargus. Phytophthora capsaicei rots away at the roots and affects the overall health of the plant. The weight of the roots were analyzed to determine below ground interactions. We found that, again, that Root Shield Plus and Stargus failed when they were combined with Grandivo. In trial 2, there were no significant differences. However, the same pattern of reduced weight when miticides were applied with fungicides. A slightly different pattern we found in trial 3 when Root Shield Plus was applied with Ancora or Grandivo. It increased root weight, but when these miticides were applied to Stargus, they decreased root weight. This may have to do with how well Root Shield Plus established in the trial. We wanted to know then to determine the actual disease severity of the roots. The root disease severity was recorded for each plant using a 1 to 5 scale based on the percentage of visible symptoms. 1 was 0%, which was a completely healthy root mass, and 5 was completely dead. In trial 1, all Stargus treatments decreased disease severity. Root Shield Plus and Ancora decreased disease severity, but when Root Shield Plus was combined with Grandivo, Root Shield Plus failed. In trial 2, Root Shield Plus reduced disease severity. When miticides were combined with fungicides, they greatly increased disease severity. In this trial, disease was relatively low in the positive control, the miticides, and the fungicides. But when the miticides and fungicides were combined, they appeared to greatly increase disease severity. We wanted to know if any of the treatments had decreased Phytophthora presence in the treatments. So 5 roots were randomly selected from each plant. They were plated on PARB V8 selective media, and colony growth was counted 3 days later. As you see here, there are the 5 roots and 1 colony growth of Phytophthora. On this other, we have 4 out of the 5. So Phytophthora was covered from all inoculated plants. We did find that there was some suppression in Root Shield Plus in trial 1, but this was not a strong trend. We also wanted to know if the fungicides were recovered in the roots of treated plants. Trichoderma was recovered in the plants treated with Root Shield Plus. It was also recovered, however, in the untreated controls in trial 1 and trial 2. This was likely due to contamination during handling. Bacillus was recovered in all the Stargus treatments. However, it was also recovered in both of the untreated controls in all the trials. It's unclear if these controls were contaminated by Stargus or from another source of Bacillus. The media used was not selective for Bacillus found in Stargus, but for all Bacillus. Our conclusions of Objective 2. Temperature may affect the efficiency of these products. Grandivo did not affect Swarovski populations in the greenhouse trials. Ancora and Grandivo reduced the efficiency of Root Shield Plus and Stargus. Grandivo did this most consistently out of both of the miticides. When Root Shield Plus was applied alone, it reduced Phytophthora root rot disease, severity, and symptoms. I do have some suggestions for improvement for future study. Inconsistent temperatures had an effect on the results. This may be mitigated by using an industrial-sized greenhouse for more consistent temperature, or the possibility of doing more replicates done at the same time of year so we can better understand how temperature affects these microbes. We originally wanted to evaluate the effects of spider mites and Phytophthora. Unfortunately, Swarovskii were not released onto the experiment, but migrated from other areas of the greenhouse. They could not be eradicated without harsh chemicals that would have prevented the infestation of spider mites. So possibly growing plants in a sterilized environment and being able to infest both spider mites and Swarovski mites would help us to better understand the effects that these microbes have on spider mites. The takeaways from my thesis. Grandivo is effective for the control of spider mites and is compatible with Swarovskii. Swarovskii are compatible with all treatments and they can be used together in an integrated pest management plan. When Ancora and Grandivo were applied in combination with Root Shield Plus and Stargus, there were consistent patterns of interference. More work is needed to understand the conditions and mechanisms these products interact with one another. My overall conclusion from my entire thesis is that at this time, we would not recommend the application of more than one microbial product at a time for the management of arthropod or pathogen pests. I would like to thank my committee, especially my wonderful advisor, Dr. Carla Adesso, who I could not have done this without her. She really is not only a mentor for my career, but also a life mentor. I would also like to thank Paul O'Neill, the head of our lab, who probably at this time knows everything as well as I do. I'd also like to thank our lab techs, Faith and Megan. And I would like to thank Dr. Fulia Basil-Gorel for letting me use her facilities and giving me an overrun with the pathogens, and especially to her lab staff, Terry and Christina, who got to show me all the ins and outs of pathogen work and have really been great teachers. I would also like to thank Dr. Jason Oliver for his support and also his lab support. And I would like to thank Dr. Lisa Alexander for being on my committee. I can't forget about the graduate students, especially Vivek, who not only had been with me through all my ups and downs, but also helped me complete my thesis after I had moved to Iowa to start my internship at Corteva. I'd like to thank Axel for being a great friend and supporting me and helping me in the lab. I'd also like to thank Milan, Farad, and Sandhya for all their help, not only being friends, but also being very helpful in the pathogen lab where I don't have much knowledge of. And last but not least, I would really like to thank my boss, Steven Zimmerman, who's been very flexible with me over my time trying to finish my thesis and also working 40 hours a week.